In Chapter 13, we talk about viruses. For most of our class, we spent a lot of time talking about cellular organisms, but our viruses in Chapter 13 are acellular, right? So these are not cells, and there's many reasons why viruses are not cells, and we will take a look at that in the beginning portion of our lecture. So we are going to separate viruses from bacteria, talk about their overall structure, and how do we identify them? How do we put them into groups when they are discovered? So what are viruses? Another name for viruses is called virion. Right? What does virion mean? This means the mature virus, the mature viral particle with its complete structure. Right? And we'll look at how different viruses are arranged. Not all viruses are alike, but they have similarities as far as how they infect cells. Remember, viruses are not cells, right? So they do interact with cells, they do infect cells, and all of our viruses are parasite, all right? So for parasite, just to review this definition, parasite means that this organism needs a host in order to reproduce, right? This is something that we talked about in other lectures, viruses being a parasite. And all of them are parasites. Again, they need a host in order to reproduce, not to feed. Viruses are not eating anything, but they need to reproduce. And this is what we mean by parasite. So these are parasites. And just again, for the word parasite, just because something is a parasite doesn't mean necessarily that it will hurt or cause harm to another cell. All right? If it's going to cause a disease in another cell, then we call that pathogen. Right, so to distinguish. So a parasite with a complete viral particle, these viral particles have nucleic acid and will have a protein coat. So that's their overall structure. So just to point out these viruses, just to give you a little diagram of what they look like, these viruses have a protein coat, and they're all surrounded by this protein structure. I'm going to draw my protein structure in blue, all right, just to, so that way we can identify this. So this will be the coat that I just drew, right? Protein coat. And then on the inside of the virus, they will have nucleic acid. All right, so I'll just put my nucleic acid as a little, let's make this black instead. All right, so that would be black. All right, so inside of this viral particle, we have this particular structure. Right, so this is what our organism, this is what our virus will look like. Okay, so again, the parasite with nucleic acid and protein coat. The virus is going to be host and range specific, but we'll come back to those points. Let's look at the composition of viruses. So all viruses have genetic material. The genetic material is DNA or RNA, right? So DNA or RNA. So let's circle this, or. This is important, right? So either can have one or the other. So this nucleic acid on the inside, the nucleic acid that we find in the inside of viruses is going to be DNA or RNA. All right, so this is what we're looking at in the inside, all right? And this area, we often call this the core of the virus, right? So this is also known as the core. And then on the outside of the virus, we have our capsid or our protein coat. So in blue, as we have drawn here, we have the protein coat. And the protein coat is made of capsomeres. So these will be the capsomeres, each of these little blue rings that I drew around my core. So it will look like that, capsomere. And when you have many capsomeres, it is then a capsid or a protein coat. So that word capsid and protein coat means the same thing. that makes up the bulk, the weight of the virus will be this protein coat. As we see here on the bottom, some viruses may have an envelope. This envelope is stolen from the phospholipids of the host. So they actually take this from the host, they steal it, and they use it, right? So how do they use this envelope? Well, by taking the envelope away from their host, they can use it as a way to go unrecognized by the host cells. They can also use the envelope in attachment. So let's say, for example, our virus is in blue, and we have, let's say we have our blue virus, and on the outside of this virus, they have an envelope, right? So where would the envelope be? It will be on the outside. All right, so let's point envelope. All right, so this is outside our capsid. So let's say, for example, we have a bacterial cell, E. coli. And E. coli now has the same color lipid membrane, right? The same factors in their lipid membrane. This is going to be the phospholipid membrane or cell membrane, right? That's in black. And the envelope here also has the same lipids. So what happens if E. coli sees this, if E. coli with its black lipid structure on the outside sees this virus now with the same black lipid structures on the outside, they're going to think it's one of them. So it makes it easier for these two structures to interact with each other. So when E. coli sees this, when E. coli cell, which is a cell, sees this virus, right, it's going to say, well, you're one of us. I'm not going to harm you, all right? So this is the virus, right? Again, it has the same lipid, we call this envelope, right? Same lipid entity as the E. coli cell, so it's easy for them to recognize each other. It's easier for these two to stick together because they have the same lipid structure. So when E. coli sees the cell, it's not going to do anything to it, and the virus is going to then be able to attach to E. coli and go in and enter, right? So it gives the virus an advantage to enter some of its whole cells if they have an envelope. Some of these envelopes may have spikes on the outside. So there are these areas that protrude, and these conspicuous areas are used for attachment. They help the virus to attach to their host cell. All right, so here goes our virus. In this picture, our virus is a little bit big, but in comparison to the E. coli cell, of course, they're a lot smaller. So maybe we should draw it like that, right? We can make this a little bit larger just for the sake of our image, so right? So E. coli would be, of course, a lot larger than the viral particle, right? But to give you an idea here. So one thing that we did not go over in the slide is what does it mean to be host-specific and range-specific? So these viruses, each virus can only interact with a certain cell, right? So you have some viruses that, for example, this virus that I drew on the board can interact with E. coli cells, right, which are bacteria. But those viruses would not be able to harm human or harm plants or harm another animal, right? So many viruses are specific for a certain host, right? It has to be a certain organism. And by range specific means usually within that host, there's many different cells. So the virus, when it enters, it cannot interact with all cells in that host, but it only could attack or attach to specific host attachment sites and cellular factors. It can only attach to and recognize certain cells within that organism. All right, so take, for example, our HIV virus, right? HIV virus doesn't attack every cell of the human, it will only attack the T cells, all right? So they are specific for damaging the T cells and weakening the immune system. But they're not going to attack all the other cells of the body, all right? So they're very specific for, again, range specific, meaning that they will only attack some of the cells, all right? Very particular cells. They're only very specific for recognizing, attaching to certain cells in that organism, in that host. So still looking at our viruses, they have three major shapes. Similar to the way how bacteria had three major shapes, so do our viruses. So what are they? The first one, polyhedral. That word means many-sided. And there's several examples that we have here, different viruses. Some of them you may recognize based on their names, some not, but we'll look at them at the end. Make sure that on your slide, poliovirus is included as one of the polyhedral viruses. The other virus that we have is the helical shape, and these often appear cylindrical or rod shape. Influenza and Ebola virus, make sure that on your slide it says Ebola, right? So those are your examples. And for the complex virus, we have our bacteriophage, which are viruses that infect bacterial cells. So even bacterial cells, as I mentioned on the last slide with E. coli, can get infected too. Any cell can get infected by viruses, no matter how small that cell is. All right, so here are our different pictures. So for the polyhedral virus, right, we have figure A. All right, so many of our viruses may look like this, and some of our animal viruses may look like this, and we have some listed here as our examples. And we can see in this figure what we're talking about as far as the structure of the virus, where on the outside we have the capsid, and this will be the core. And depending on our virus, the nucleic acid, remember, can be what? It's either going to be DNA or RNA, all right? So let's say if we had something like herpes virus, the herpes virus is going to be a DNA virus, all right? But something like your rhabdovirus is going to be an RNA virus. Okay, so here we have it. So what is in the middle as far as the core is going to vary with different viruses. Here's a picture of our helical virus. As I mentioned, it looks more cylindrical shape. Again, the capsid is on the outside. The capsid is made up of many capsomeres. Each dot or each unit here is going to be the capsomere. And then on the inside, we have our nucleic acid, right? So the nucleic acid could be DNA or RNA, varying for different viruses. So this one, again, the helical will be here in the middle. And for our complex viruses, many of our bacteriophages will look something like this. Here we have the representation of the T-even bacteriophage. T-even is usually a way to identify and classify these viruses. So we have, for example, T2, T4, T6, all right, following this even scale. And inside of the head of the virus, we have the DNA, or it could be RNA. So the genetic material, the nucleic acid is here, all right? So this is where the nucleic acid will be, right, in the head. So what else is in this virus, all right? What are the other parts? There is the sheath, and the sheath contracts to inject the genetic material into the bacterial cell, right? So the genetic material is here. The genetic material will have to go down and down and down, and it will go out through this area to where this virus will be attached to a bacterial cell. So let's say if we have our bacteria, let's say this is E. coli. E. coli gets infected by many of these T-even phages, all right? So this will be the pin in the base plates at the bottom, right? And then you would have this polyhedral head at the top that will have all the genetic material, all right? So in order for the genetic material to get from the head and down, all right, into E. coli, the sheath is going to contract, all right? So at the bottom of the complex virus, there's tail fibers, pin pin and the base plate, and this helps for attachment to the bacterial cell. So again, the tail fiber, pin, and the base plate. These are for attachment. All right? So I told you guys before what the sheath is for. What is the sheath for? So the sheath is for injecting the DNA into the host. Okay, so this is the mechanism that the even phages will use. Our viruses, when they're discovered, they're going to also be classified into their own taxonomy grouping. And the family names end in viridae, genus names end in virus. So this shows you, gives you an example here, where for herpes virus, herpes viridae, all right, the ending in viridae indicates the family name. And we know that that grouping is going to come before our genus name. So we'll put here family name. Let's do it here maybe. All right, so this will be the family name. Right? And then for herpes virus, all right, this will be the genus name. All right, so when we see virus at the end, we're talking about the genus name. So Viridae family, AE, right? Virus, all right, will indicate the genus name. And when we're talking about species, oftentimes we might identify it with the host, all right? So species, humans, herpes virus, all right? We're identifying that it's going to infect humans, all right? And then for subspecies, sometimes they are labeled as 1, 2, 3, all right? Or 4, with the herpes virus, it goes all the way up until about 8, all right? So these will be the subspecies. And over here, similar with our retroviruses, this gives you an example. For HIV virus, Retroviridae will be the family name. Lentivirus is a series of viruses that they are together with. And the human immunodeficiency virus, all right, that's the species name. When we're talking about HIV-1, HIV-2, that will be the subspecies. All right, so subspecies are designated with a number or letter. And in the case of herpes virus and our lentivirus, these are indicated with numbers. Can you think of any viruses that have letters at the end? So what about hepatitis, right? Hepatitis A, B, C, D, those have letters at the end, right? So some of the viruses, they have, as the subspecies, it will be a number or letter. Here we have numbers, but sometimes it can be a letter. Right? So this is just to give you some examples. So our viruses were first viewed with our electron microscope, and these microscopes are very expensive. We do not have them in our laboratory. They cost hundreds and thousands of dollars. But the electron microscope, we do have 2 on Lehman College campus, and the electron microscopes, they will allow us to magnify our structures a lot larger than our light microscopes that we use in the lab. So the electron microscopes have allowed us to magnify images up to 100,000 times its size, all right? So we can make these small viruses that are a lot smaller than the bacterial cells, we can make them a lot larger so that way we can see them, right? And we, of course, do not get to see them in the lab, but they do interact with humans, so we discuss them here in microbiology. So let's talk about the differences between bacteria and viruses. And if you're not sure about some of these structures of bacterial cells, you can always go back in the book or in other past lectures to review the structures of bacterial cells. So for bacterial cells, most bacterial cells are not parasites. There are some exceptions. So for this, you need to go back, all right? So what are the exceptions? I'm not going to give them to you, but in class, I will ask you about them. So what are your bacterial parasites? This means you need to go back to chapter 11 to figure out what are these bacterial parasites. So there are a few of them that we've studied. Which ones are they? If you don't remember, you have to go back to that lecture on chapter 11. Okay, so viruses, they're all parasites. So yes, all of them are parasites. And again, this word parasites, do you remember what it means? So this means, again, it needs a host to reproduce. Okay, so as far as plasma membrane, our bacteria are cells, so they do have this plasma membrane. Viruses, they're not cells, no plasma membrane, right? So we do not see that structure. Some of the viruses may acquire, or as I mentioned, steal an envelope, a lipid structure from the whole cell that is a part of this plasma membrane, or looks similar to this plasma membrane, but it is not the plasma membrane. Okay, so no. Binary fission, we see this as a reproductive mechanism in bacterial cells, but viruses do not use that mechanism. As far as their size, right? So most bacterial cells, we measure them in micrometers, where our viruses, we measure them in nanometers, right? Smaller, nanometers. So this is micro, this symbol, all right? I put it, the U here stands for micro. Micrometer, right? The N, nanometer. So nano, right? I'll just put nano here, right? So comparing micro to nano, right? These structures are 1,000 times smaller, right? Very, very tiny. So when we discuss their size, we use the guide, do they pass through the bacteriological membrane filters? And these membrane filters, again, if you're not sure about what they are, you should go back to your notes in chapter 7, right? Where we talked about membrane filters. What is the size? This is a good question. What is the size of these? What is the pore size? Right? What is the pore size of these filters? Right? So they pass through the bacteriological membrane filters. For bacteria, they can't pass through the filters, the pore size, but our viruses can. So what is the pore size? You guys need to go back. I'll have a question for you guys on your handouts in class. So for your bacteria, they possess both DNA and RNA, right? DNA, we've talked about this structure in replication, is also required in expression. RNA is required in both as well too, in bacteria or any cellular structures. Viruses, it has to be either DNA or, right? So DNA or RNA, right? So not both, all right? Either you have DNA viruses or RNA viruses. Do they produce energy? Do they have a mechanism to make energy? Can they metabolize? Are they making enzymes for metabolizing substances? For your bacteria, yes. For viruses, no, right? So in order to get certain enzymes, to get energy, your viruses need the whole cell. They require the whole cell in order to do that, or to get those things. Ribosomes. Bacteria have ribosomes, and some of you guys still don't know the size of the ribosomes. All right, so what are the sizes? You guys need to go back. What are the sizes of these ribosomes? We have ribosomes in bacterial cells, and also in eukaryotic cells. What are the sizes? What is the small, the large subunit? What is the complete structure? You need to know this. This is something that we learned from the first exam. So the bacterial cells, they have the ribosomes, and for your viruses, no ribosomes. So if the virus wants to synthesize a protein, if they need enzymes, they need to get that from the whole cell, because they can't make it on their own. Right? So these are some reasons why the virus needs the whole cells, because certain things they can't do without it, right? And they need them in order to reproduce. And when we're talking about reproduction, and especially making DNA or RNA that the virus needs, you need enzymes to do that, right? So without the host cell, the virus cannot make those enzymes. Sensitivity to antibiotics, bacteria we learned are sensitive to antibiotics, but viruses not. All right, so if you have a viral infection, let's say if you go to the doctor with, let's say, the flu virus or cold virus, they will send you home. Right, so antibiotics are used mostly to treat bacteria. Some fungi will be treated with our antibiotics. Okay. So now we're going to talk about how animal viruses are grown. How do we grow them in the laboratory? For one, we cannot use nutrient agar. Broth or agar, or glucose broth or agar, which we discussed in growing bacterial cells. Okay, so how do we grow them? In order to grow viruses, we need host cells, right? We have to provide them with their host cells, their specific host cells in order for them to reproduce. So one way to do this is by growing them in an embryonated egg, right? An egg that has living cells, all right? So that we either grow it in an egg or you grow it in a live animal. And when the virus grows, what will happen to this living organism? Well, they're going to show some sort of sign that they've been infected. So either the egg or this growing embryo will die, or it will get sick, or maybe it might present some sort of tumors, but it will show some signs that it has been infected. And from there, we can, once the virus is growing inside and we see these signs, we can extract these viruses to study them. We can also extract them to make things like vaccines, all right? So this is one way to grow your virus. So again, in order to grow viruses, you need host cells. So you cannot use nutrient broth or glucose broth or their agar constituents in order to grow the viruses, because inside of these there's no host cells, so this will not work. Okay, so if we use the actual embryonated egg or animal, we can see signs when the viruses grow, and the sign is that they either become unhealthy or sick. They might present different signs, or sometimes they die. But that's all going to be an idea that you have the growth of a virus. So they're showing you here, they're injecting different sites of the egg. If it's a hard egg, they may have the drill sites in order to inject the virus, but they're showing you how it's done here, right? Today we use cell cultures in order to grow our viruses, right? So what are cell cultures? The cell cultures are going to be similar to or the way how it's presented here. We're growing our viruses, but we're growing them with whole cells. So cell cultures have whole cells, all right? So whole cells are present. All right, so this will allow our viruses to grow. So how do we get the cells? We get the tissue that we want, right? We make sure that it's specific so that way that virus can infect those cells. The tissues are broken up, all right? So remember, if we have tissues, we can break them down into cells, which will be smaller. So this is larger, and these will be, of course, smaller. And then from there, we can put it, suspend it in a petri dish. With nutrients, right? Culture medium. Remember, medium means nutrient, right? and then what will happen over time, the cells will grow, right? So now we have an environment for the viruses to grow, right? Whatever cells we want, again, we take whatever tissue, right? We get the cells, we break them up, we then put them in the petri dish with nutrients so that way the cells are healthy and they're growing. And then what we do is that then we can inject the viruses, all right? So let's say this is a little needle, right? We will inject the viruses. Right, so when we inject the virus, what happens? Then we look for something, we wait. And when we see a sign, we know something is wrong. So there's different ways to detect this. Normal cells, as they show you here, they grow in a flat layer. And sometimes when we inject our cells, our whole cells with viruses, we can see the transformed appearance. And transformed appearance represents an abnormal growth. Right, so here they explain to you normal cells grow in a uniform manner where they're usually flat in a thin layer, but in the case of transformed cells, they're more or less like tumor-like, and they grow with no inhibition. They're growing everywhere rapidly. So you have this sort of overgrowth and abnormal appearance of cells. They are not growing in a monolayer. So what are some advantages to growing cells in a cell culture as opposed to growing them in animals or in eggs, all right? So there's 4 benefits of using the cell culture. 1, we can still detect the growth of the virus. And remember, when we are detecting the growth of the virus, we are looking for a sign, right? So what is the sign? These are 2 signs, all right? So either we may see plaques, and what are plaques? When we inject the virus, so remember here we inject the actual virus so that way they can infect these whole cells and we'll see something. So the way how this will look, just to give you a picture of this, I'll go to the next screen, and you guys do not have this picture. But what the plaques look like, if you could see it here, is that so if we didn't have any virus, we would see something like this, all right? So no virus injected, or the virus didn't infect here. But in this case, what we'll see here, if the virus has infected the cells, we'll see these little plaques. And the plaques are going to be these clear areas, so P-L-A-Q-U, plaques, E-S. All right, so they'll look like these, almost like the zone of inhibition, where you see these clear areas. And these clear areas represent where the host lysed the bacterial cell. All right, so I pointed to them. You see several of these little plaques here, all right, and those will all represent where the virus has infected. They get into the actual bacterial cells and they damage them. All right, so what you see are these plaques, the clear areas. All right, so you can again see the signs. All right, the other sign that we can see is a transformed cell, and I gave you guys that example up here where you have the abnormal growth, right? The cells do not grow as normal. So you look for those signs, and from there you can extract the viruses and study them, and to look at them, right, in a laboratory. Using the cell culture, we do not harm animal cells, right? So we're not killing any animal cells. In this way, when we use an embryonated egg or a live animal like a rat or a rabbit or a chicken or any animal, right, we're harming animals. So if there's some sort of ethical concerns, that we're killing organisms in our studies. In this case, using cell cultures, we avoid that. Number 3, right? So cell cultures allow us to study certain human viruses, right? So example here, HIV, right? In the case of HIV, for HIV, this requires human cells, right? Human cells and certain human cells in order for us to see the signs, how they truly infect, right? If we infect a rat or a chicken with HIV virus, we won't be able to truly study the way how this virus infects humans, right? We see the HIV virus is host specific for humans, right? That's where they cause their infection. And the other thing here as the benefit to using cell cultures is that they avoid allergies, right? So if we grow the virus in a cell culture in its natural host, and then we extract those viruses in order to make the vaccine, then we're avoiding animal allergies. If we grow the virus in an egg, some people may be allergic to eggs. So when we extract the virus from that environment of the egg, certain people may be allergic to those vaccines that are being made. So to give us an idea of how these viruses grow in our host cells, we're going to look at the multiplication of the DNA containing animal virus. So how do they infect? Right. So this is our diagram from the book to go over how they infect cells, and you can also watch this video. You can copy and paste the link to get an overview of how these viruses infect cells. So in looking at this, where do we start? So what's most important right here? Right, this is the most important step of the infection process of viruses. And why is this most important? This allows the virus to get inside the host cell. So what step is this? This is attachment, right? And in this attachment, the virus has to attach to its host cell, right? This is the first step in the infection process, right? So that way the virus can replicate. And remember, if some viruses have an envelope, it makes it easier for them to go unnoticed by their host cell, right? And giving them the advantage to attach. So step one is attachment. So we have here goes our virus, right? This is a DNA virus, right? We see the capsid, the protein coat, and this virus attaches. They have to stick together, right? So at one point, this host cell is saying, okay, we're okay with this virus, right? We do not have any problem with this virus, right? We're friends with this virus, all right? And that's until the virus comes inside, right? So it's almost as if a stranger comes to your door, you open up the door, and you say, okay, well, this person looks friendly, but the minute you let them in, right, they start stealing everything, they break everything, they maybe try to attack you. You did not think that they would do all that stuff when you saw them at the door, all right? So this is what's going on with your viruses. At this recognition point of attachment, they see this, the host cell sees this virus, and they're saying, okay, you look like one of us, right? So this is where we're at. So in the second step of the viruses, there will be entry, right? So they get in, right? So the actual virus, the host lets the virus in, all right? So again, going back to meeting this stranger at a door, you say, okay, you look friendly, you let them in, right? But you don't know the damage that this person would do once you let them in the door. And the same thing with the viruses, this host cell doesn't know what they're up against. So entry, we'll put this as 2A. Right? So how does entry occur? Entry occurs by endocytosis, and endocytosis is when the host will eat the virus. So they actually take it in, right? They'll bring it in, right? Not in a way to harm the virus, but they'll bring it inside, right? And fusion, right, is a second way that this can be done, where fusion is often seen with envelope viruses, where the envelope, since it's similar in material to the whole cell membrane, it will mend together, right? So it sort of blends together, and then the virus comes inside. And then we'll put this step as 2B. So what happens in 2B? Uncoating, right? So once the virus comes inside, it will lose its protein coat, right? So all the capsomeres are degraded, right? So here we have our viral particle. So this was the virus with the capsid. Now it has lost its capsid. So it can either be degraded by the viral or host enzymes, right? So when the virus comes in, it is in the cytoplasm. In the cytoplasm, this is where it loses the capsid. And once it loses the capsid, right, the capsid is really to shield and protect it as it travels or moves around from one host cell, organism to the next. But once it's inside the cell and it's getting ready to replicate, it doesn't need this capsid any longer, right? What's most important is its genetic material. And that genetic material gives all the instructions. Remember this, the DNA is the control center for these particles, and for, if we're talking about organisms, cellular structures, right? DNA is the most important structure, and this is what it needs in order to replicate. So the actual DNA, right, where does it go? It's going to enter into the nucleus, right? Why is it so important for the DNA virus, for the DNA, right, the DNA of the virus, so we'll call it the viral DNA, to enter the nucleus of the host? Right, so why is it important for this genetic material to enter, genetic material of the virus to enter the nucleus of the host? Well, one thing is that it wants to get to the control center of the cell. So once it gets to the control center of the cell, it can, one, shut down everything that is going on in this whole cell, right? So anything that the host is trying to do, it will block it, inhibit it. And the other thing that's important is that the DNA, in order to be expressed, in order for this viral DNA to be expressed into RNA, it has to get inside of the nucleus, all right? So inside the nucleus, this is where the viral DNA will be made into RNA, right? So the expression, transcription, right? This is something that we discussed. So the viral DNA needs to get inside, right? And once the viral DNA is inside the nucleus, again, it can be expressed, right, into RNA. So now that the viral genetic material is inside of this whole cell nucleus, now it's taking over, right? Think of this almost like as an alien, right? Taking over, right? They want to get to the control center, the command center, in order to shut everything down. So in our next step of our viral infection, the virus is going to express its early genes, all right? So what are the early viral genes? As indicated here, many of them include the virus's DNA polymerase, right? So not any DNA polymerase, the virus's DNA polymerase. And that DNA polymerase will allow the virus to multiply, right? It allows the replication of the DNA, right? So it has to express the DNA polymerase, right? And it does this by getting into the nucleus, expressing the DNA into RNA, and now it can start to make its viral, express its viral gene and make the actual enzymes that it needs to copy its DNA. All right, so here is we said that this was the actual DNA, right? The DNA was then made into the RNA, right? So we said it was expressed. And in this case, once this is done, then the actual virus can be replicated. Right? So now we go from one little virus, right, particle, now we have three, right? So we have the DNA polymerase that is made, and in order to, the DNA polymerase is made in order to replicate, make many copies of this DNA for the virus. So once this is done, the next set of genes that is expressed in the viruses, now we have multiple viruses genome will be the late genes. And the expression of the late genes includes the genes that are responsible for making the capsomere proteins. So this now allows the virus to assemble itself so that way it can exit, right? So here are the capsid proteins. All the capsid proteins are made here, and these capsid proteins eventually, in some cases, the capsid proteins, and it's different for different viruses, the capsid proteins have to get inside of the nucleus where the virus is assembled, right? So in step 6, we have maturation, where we have assembly of our viral particles. They will now put the capsomere proteins on its outside, so that way it's ready to exit the cell, right? So in this case, they're getting ready to leave, all right? So step 7, we have the release of our virus, and it's ready to go on to the next organism or the next cell. So when these viruses are released, different viruses are released in different ways. Right. So I did update this slide a little bit. Envelope viruses, they bud off, right? So as they're budding off, they're pinching part of this plasma membrane, and some viruses will take the plasma membrane and make it into its envelope. In this way, so we can see this here, if we look, so this is pointing, I pointed this arrow here to the budding. We can see this helical virus, again, it's pinching, and eventually, notice how when it's released, it has an envelope, right? The yellow portion is the envelope that looks similar to the whole cell membrane, right? So the whole cell membrane would be this, right? So the whole cell plasma membrane. So they pinch it, they take, and again, this helps when the virus goes on to recognize or attach to a similar whole cell, right? When they bud off, the whole cell remains intact, right? So they are safe. For non-envelope viruses, when they leave their whole cells, they will kill it. So the whole cells are lysed, right? So they're damaged, they're dead, they will not come back, right? So it's 2 different exit strategies. And just because the envelope virus is not, the actual membrane is not harmed, it doesn't mean that the actual cell is still a viable cell, or it doesn't mean that the cell is going to be the whole cell is going to be functioning as it did prior to the viral infection. More than likely, it's harmed or damaged in some other way. So let's look at some of our viruses and some of the terminologies here, right? So I'm not sure for your slides, it may be slightly different, but I did change it here so that way we focus on all of the DNA viruses first. And then for the last 2 slides, it will be all RNA viruses. So the first one that we have is in the Adenoviridae family. And this is your adenovirus. The adenovirus usually cause several respiratory tract infections. Many of them may even mimic the cold. It may also cause things like bronchitis, right? You can see many symptoms when the adenovirus infects humans. These are, again, all animal or human viruses. And it has a polyhedral shape, no envelope, right? So the blue indicates the capsid, right? If we see something like yellow, that means envelope. So there's no envelope. Adenoviruses are what we call persistent viruses, and persistent viruses, they may occur for several years, meaning that they may stay in the body for several years, especially if your immune system doesn't attack it quick enough. And if it's not treated, or if your immune system doesn't deal with it appropriately or efficiently, then it can kill you. The herpes viridae, what is in this particular family? Well, we have all of our herpes viruses, and herpes viruses 1 through 8. And these cause infection, a number of infections caused by the herpes viruses. We have cold sores, chickenpox, shingles, mononucleosis. We'll talk about this in our last couple of slides before the exam. Several lymphomas, right? So these are tumor infection. Kaposi sarcoma, right? Another tumor-like infection. These are envelope viruses, right? So the yellow, remember, means envelope, and oftentimes they are latent, right? Latent means that they remain in the body. Some of them are dormant and asymptomatic for a period of time, where you wouldn't notice that they're present in the body, but then they can reappear, and the signs and symptoms reappear once the virus comes out of its dormancy. Right, so these are viruses. For the adenovirus, we will call this a DNA non-envelope virus. Right, so DNA non-envelope virus. Right, but something like our herpes virus, this has an envelope. So how do we call it? This will be a DNA envelope virus. Right, so these are all DNA viruses that, and remember that I said the herpes virus, some can be, we'll just add in here, some cause cancer or tumors. All right, so we'll add that. Okay, so we have a few other DNA viruses. Right, so in the Hepnaviridae family, we have our hepatitis B virus. Right, so hepatitis B virus is a DNA envelope virus. It has polyhedral shape and it is persistent. Right, anybody know what hepatitis, or what area of the body hepatitis infects? What cells? All right, so hepatitis, this will infect the liver cells. And your hepatitis B virus, this is the only hepatitis virus that is a DNA virus, right? All the other hepatitis viruses, hepatitis A, C, those are going to be RNA. So we'll see those in our RNA slides at the end, right? We're coming to those. All right, so these are persistent, meaning that once they infect the liver cells, if the body doesn't deal with it or you don't get medication, you can die, all right? It can be fatal. So our next DNA virus is the Papoviridae, and the Papoviridae includes your HPV, human papillomavirus. This is a DNA non-envelope virus, and it is cancerous, meaning that tumors will form, right? So tumor formation. The Poxviridae family, we'll talk about this in more detail. Many of these viruses we'll talk about in more detail during class time. But the Poxviridae includes your smallpox. This has an unusual shape. It is a brick-shaped virus. All right, so this is the brick-shaped virus, and it is persistent. All right, so this is a DNA envelope virus, and we'll talk about smallpox when we talk, or you guys can review before Edward Jenner. So you guys can review, who is Edward Jenner? All right, you guys can look at that before I get to it. Now he's discussed in chapter 1. Okay. So now to our RNA viruses. So the RNA viruses, there's a number of them. Some of them which we are familiar with, and the Filoviridae family, Ebola. Right, Ebola causes a profuse bleeding, and this is a helical RNA envelope virus, right? If you as we've seen in the news in recent times, we have seen Ebola where it has killed many people, right? It's persistent, right? If your body cannot deal with this, or you're not taking any medications to help, it is fatal. The Orthomyxiviridae family, this includes your flu, right? So this is your influenza, this is the flu, right? The flu virus. It is a helical envelope virus, and it is persistent, right? The flu has killed people, right? It's fatal. It could be fatal if it goes untreated or your immune system doesn't attack it. You have the Rhabdoviridae, and this includes your rabies virus, right? The rabies virus is a bullet-shaped virus. It's helical, but we give it a bullet shape because that's what it pretty much looks like. All right. So this is an envelope helical virus, and our last envelope virus on this slide in the Retroviridae family, we have HIV virus 1 and 2. Right, so this is more or less the subspecies name. Okay, and the HIV virus, this is an RNA virus, and it is polyhedral. Some are persistent, some cancerous, and some latent, right? Very special virus. It can take on many forms, and once it infects humans, it will interfere with their immune system. So our last RNA viruses are on this slide. All right, so in the Pyrrhocornviridae family, we have hepatitis A, and hepatitis A is included here. We also have our cold virus, rhinovirus. It causes greater than 50% of cold infections. All right, these are our RNA polyhedral virus. All right, in your Hepaviridae family, we have our hepatitis E virus, and hepatitis E also polyhedral shape. The Flaviviridae family, this is now your hepatitis C, right? And this is an envelope polyhedral virus. And then in the Delta Viridae family, we have hepatitis D, which also has an unusual shape. Make sure on your slides you update it to round. All right, so it is a round non-envelope virus. So most of the hepatitis viruses, as I mentioned, they are RNA viruses. Okay. So this concludes our viral lecture. And for your viruses, similar to the bacterial cells, you would need to know their overall characteristics. So make sure you know the characteristics of these viruses. We will talk about them in greater detail soon. And if you're still stuck or have not familiarized yourself with our bacterial characteristics, it's a good idea to go back and start going over them and learning them now.